Hi, everyone. This is episode 733 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today I am excited to be joined by our guest, Dr. Susie Dai. This episode is made possible with support from our sponsor, New England Biolabs. Listeners, are you curious how molecular biology is moving forward around the world? Hear from the scientists who are driving discovery and expanding applications of existing technologies on the New England Biolabs podcast, Lessons from Lab and Life. Host Lydia Morrison interviews revered scientists working in genetics, synthetic biology, diagnostics, molecular cloning, and other fields. Get ready to explore the cutting edge of science and learn what these remarkable scientists have to offer. Don't miss out on the opportunity to hear exclusive stories about the world of laboratory research. Tune in to Lessons from Lab and Life today, wherever you listen to podcasts. And today, our guest, Susie, will share stories from her own life and research. So listeners, get ready to meet another one of our outstanding people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am excited to be speaking with our guest scientist, Dr. Susie Dai. So, Susie, welcome to our show today. How are you?
I'm doing good. Thanks for inviting me.
Well, Susie, I am thrilled to have you here with us today, and I'm looking forward to learning more about you and the work that you do. But let me first give our listeners a little bit of background. So listeners, Susie is Associate Professor in the Department of Plant Pathology and Microbiology at Texas A&M University. She is also Director of the Biomonitoring Program in the Iowa State Hygienic Laboratory, leading Iowa Statewide Biomonitoring Public Health Surveillance. Susie received her bachelor's degree in chemistry from Fudan University and her PhD in chemistry from Duke University. Afterwards, she conducted postdoctoral research with the Scripps Research Institute and Oak Ridge National Laboratory. Next, Susie began working as a research assistant professor and was later promoted to research associate professor at Texas A&M and the office of the Texas State Chemist. Subsequently, she served as associate director of the Iowa State Hygienic Laboratory at the University of Iowa and director of the Environmental Health Division. She returned to Texas A&M faculty in 2019. And Susie, today we're excited to get to know you, of course, as a scientist, but also as a person. So can you start by telling us what you like to do when you're not doing science?
I like to read, and that's the primary thing, and just kids in the yard.
Oh, wonderful. Well, tell us a little bit more about your kids.
Yeah, I have two daughters, and they're wonderful daughters, but they're coming to the teenager, and it's a challenging time.
Wonderful. And you mentioned reading as well. We love talking about books on our show. So do you have a favorite book or perhaps one that you've read recently that you'd like to recommend for us, whether it's related to science or not?
Yeah, that's wonderful because actually I'm getting to the books by Smil, which is actually a scientific writer, introduced a lot about energy and how our daily life works in the scientific world and in very plain language. I would recommend books from him.
Wonderful. We will add this author to our website for our listeners to find there if they're looking for a fantastic scientific related read. And we talked about life outside of science. I'd love to talk about your research as well, Susie. I know you're doing phenomenal work in the lab. So how do you describe your work, sort of the big picture of what you do, to someone who's not in science or not familiar with your particular field?
Yeah, that's a great question. So I am a chemist by training. So I approach questions and real-world issues with what I have been training for many, many years. When I came back to Texas A&M, I have some interesting experience working with people, communities, and regulatory agencies. So I make up my mind. I want to build a research program that's built on the interface with chemistry and biology, which I deeply believe sustainability is coming from a natural process to synthesize and also to deal with those chemicals. So some very important or challenging issues our modern society is facing now, how we can have a sustainable production of different chemicals in our modern society, and then how we can deal with the fate of those chemicals we have synthesized, which are very stable in our society. Those are actually two sides of the equation, but they're integrated. If you think of the life cycle of how chemicals are synthesized and how they are used and how they're disposed. So that is why the center and more kind of philosophical question I'm trying to ask myself, how can I use microorganisms to synthesize a chemical, or how I can use microorganisms to degrade a persistent chemical. That's kind of the theme of my research direction currently in my lab. And besides that, I'm still looking into the interface of biology and chemistry. And especially when you think of those chemicals in the environment, then our body as human beings are exposed to that from via different routes. So that's another kind of project I do in my lab is biomonitoring. So we try to study the human population and find out their exposure content and burden to those toxic chemicals in the environment.
Very cool. I love these two different sides of the work that you do, both kind of getting these microorganisms to work for us to produce things that we need, and then also just this broad field of biomonitoring and examining what's in the environment and what humans are being exposed to. I think these are two very cool areas. So I know in science, the work that you do can be inherently motivating, and we love talking about motivation and inspiration on our show. So Susie, do you have a favorite quote or a saying or something in particular that really keeps you motivated, whether it's related to science or not?
I have a favorite quote, and not forget it. I kind of feel nervous. So I like chance favors the prepared mind. That's from, I think, the greatest combination hybrid of chemist and a biologist.
Absolutely. So how does this apply to your everyday life?
When I'm planning for something, and I will try to think, what are the conditions I have so I can implement my plan? And then to get to there, what are the things I have to learn? What are the resources I have to acquire? And what are the people I have to know? And what are the team I have to build? So those all kind of come together, even with a research project. So let's say I want to fix this problem for this persistent chemical. What's the available technology and what I can do very uniquely into this field? Then I like the quote, chance favors the prepared mind. I just need to try my best to work as a sponge and then absorb the knowledge that's coming to me. Of course, get rid of the misleading information as much as I can, and then make the most important decision of get the resource, work with the right people, and build the correct team and fix the problem.
Absolutely. And I love thinking about this idea of you just sort of computing all these different paths and what different things need to be done along the way to make sure you get to your goal. And I think getting to your goal, not only having plans is important, but sometimes having the right people in your life and in place is important. So Susie, when you look back at your scientific career and at your life, maybe more in general, who was a role model or someone who really had a big impact on you during your career?
I don't think I have a single one. I want to learn from people around me. And I want to be honest, every people have to have a piece of pride in themselves. They can do things well. And at the same time, they have to be very humble because none of us can do everything by ourselves. So I learned from each boss I worked with before, from my PhD advisor to my postdoc advisor, then to the boss I have to work with in the past in different positions. And each one actually teach me something different because they have different personality, they have different skill sets, they have different perspectives for life and for the career. And I learn from each one deeply.
I love that. I love kind of picking up all these little pieces along the way from the different people that you've interacted with, the different leaders that you've worked with in these groups over the years. So let's talk about your career path next. I think every scientist gets to where they are today through a different path, some of them more circuitous, some of them more direct. So let's talk about where your journey began, Susie. When did you first start getting interested in science and start maybe thinking about it as a career?
I get my early interest in science when I was in high school. But think of the career, how I'm going to apply the things I learned is much later because I keep changing my path, even though I never changed that thing. When I was in high school, I was in the class that we were able to have some advanced teachers came to the classroom and teach us about physics and chemistry. So I got very interested in chemistry at that time, knowing all those hybrid orbitals about the atoms and other things. I think that's very interesting. I want to start this when I get into college. Of course, I found out later when I'm really in college, I started something totally different from what my impression was in high school.
Absolutely. And then when you were in college, did you start thinking about what are the potential career paths, or how did you start getting interested in the laboratory side of science and doing research as a career?
And that's a very interesting question because I want to be very honest. I'm not planned at that time. When I was in the chemistry department, I was trying to learn those things. And then I got my degree in China. And I know a lot of people are going to the United States and seek a PhD, more advanced degree. Then I was kind of follow the crowd. Okay, I know that's a place with all those wonderful technology and the best training programs. I want to go there. So that's what I did when I was in college.
And you ended up at a wonderful school, Duke University. What was your experience like there?
Yeah, I love all those people around me. And also we have different professors with different research background. Then we have the chance to look into which one you want to work with. And I'm very fortunate to have this mentor who actually helped me grow a lot, not only in the discipline of chemistry, but also in writing, in presentation, and to develop a systematic mindset to do your research project. And over there, that's also kind of get back to your early question about your career interests. Again, at that time, I was still very young. And I think this is a common experience with many young students at still our age right now. When I was in graduate school in the first 1 or 2 or 3 years, I would think, I will work for industry. Because if you're a chemist, it's not hard to find an industry job, and it's usually highly paid. And then I had that mindset in my graduate study, and I was given the opportunity to go to those national conferences because my mentor supported us to go and present our work. Then I met with some colleagues that they have worked in the field for many years. They're working in the industry. Then I was thinking, okay, if I get an English job like this, and then after 20 years, I'll be like those people in this conference. Do I really want to be like that? After going to the conference for a couple of years, more in the senior time of my graduate study, I decided I probably want to stay in the academia because I can be more open with what I want to do in terms of scientific questions. Because in industry, you don't make those decisions easy, or driven by the economic gain of the company or the group priority to make sure the company can be successful in the market. So that's kind of a turning point, because I know I can always go back to industry if I have that opportunity or if I want to in the future.
Absolutely. So it sounds like you started to feel the value of being able to have this freedom in answering the questions that you want to ask. Then how did you ultimately choose where you were going to do your postdoctoral work?
So the first postdoc work was actually when I was in the conference, I was about to graduate. So I give presentations and meet with different people. I would think which group I want to do my study. And this group was in Florida. Then they have a new site sponsored by the state together with other investment. And they have a brand new drug discovery group. So at that time, I was very interested to know how people come up with all those like pipelines and developing a drug to treat a disease. So that's more kind of a topic driven. So I chose to work as a postdoc in that group, which I was surrounded by so many wonderful scientists coming from GSK. If you don't know, it's like one of the biggest pharmaceutical companies back at the time. Eli Lilly, Merck, and those are still pretty huge pharmaceutical companies present at a global scale. I was very fortunate to learn from different people with different disciplines and find out in those pipelines, many people with different backgrounds have to work together to produce one drug. So that's amazing environment and atmosphere. I learned a lot from that interaction, together with understanding how the industry works, together with how the fundamental research contributes significantly to those drug mechanisms, together with product development.
Absolutely. And I think it's really cool that you were able to get some of this industry exposure through your postdoctoral work. I think sort of being able to see both sides of the equation helps you make an informed decision about what path you ultimately want to pursue going forward.
Exactly.
And then you decided to go for the faculty route. So what was that experience or that transition like for you, Susie?
At that time, actually, there is a family reason that my husband is actually in Tennessee. So again, I'm in that field that I learned about this industry, and I'm also kind of moving ready for another interesting platform I hope to learn as well. So I'm a chemist, and that's how I went to another technology platform. It's called proteomics. So I moved into the Oak Ridge, did my second postdoc. Then I moved into this kind of hybrid position in Texas A&M, which is a state agency housed on a university campus.
Yeah. So you've had exposure on all sides. You had sort of the government state lab experience, the industry exposure, at least, and then the academic position. What ultimately, I guess, tipped the scales to make you want to pursue this pure academic route going forward?
Yeah. And that's a period of about 10 years. It's a combination of both service and research. Together with a small component of teaching, because you're on the university campus, you're always invited to get lectures and also talk to students. So over that 10 years, the first 7 years, I was working in the Texas State Agency on a scientific team and also kind of leadership, all those activities in the scientific analysis world. And where our agency regulates animal feed and fertilizer. So it's a huge industry if you think of the agriculture status of many states in the United States. And then over there, I get an idea of how when regulatory agencies have to make a decision, what are the standards to back up that decision? And then chemistry, particularly in this discipline, plays a very important role for decision making, especially for quantitative and analytical results. So over there, I moved to a more kind of agency with a much broader scope in Iowa that supports not only regulatory, but also for other surveillance purposes, such as Department of Health, such as Department of Natural Resources, which are the equivalent like CDC and EPA on the state level. Then over there, I learned more things than chemistry in feed and fertilizer and in food. So I learned all those chemicals in the environment, which leading to much bigger kind of conflicts or tension between who is producing that and who is a victim of exposure to those. So I spent about 3 years in there, and of course, understanding the exposure potential health impact to human being. That's why I'm still running those campaigns for biomonitoring. Then it drove me to think, if I'm going to come back as a faculty member, I want to come up with research projects and programs that can find solutions to balance those conflicts in our society. So I think that's kind of finished the loop of who I am today.
Absolutely. And I think one of the really cool things about your career path is this idea that your training was originally in chemistry, and you started incorporating or perhaps building a bridge between biology and chemistry then kind of in your postdoctoral stages, and just sort of kept building on it at each stage along the way. So was this difficult for you initially, starting to speak the biology language or incorporating that in with the chemistry that you were doing?
Oh, it's hard. It's still very hard for me today. I cannot pronounce Latin, and many of those bacteria and fungus names are in Latin, so I cannot pronounce them. I think, okay, number one, number two, and use those abbreviations. But that's not the thing stop us from thinking and work with our people, and that's not the thing to stop me from learning about things behind the names. It's the same thing. If you call somebody and then you know that person by calling that name, but actually I learn about the personality and activities that person is doing. So what I'm trying to still is learning from my colleagues, even learn from my postdocs. Because then when I build a team, then we talk and we communicate, and people contribute on things I don't know, and I have perspectives that other people don't know. And then I have chemistry background, and biologists don't know. When we talk together, we can always have like a brainstorm and great ideas coming out. So one of the very fundamental driving before I even come back to A&M is some of the research projects I did. If I move to Iowa, like we work with different organisms at that time, more looking into fundamental mechanism, how this works, how that's not working. But I know like certain fungus and bacteria can do degradation. And when I was in Iowa, I say, hi, we have such a huge chemical contamination problem in water, in environment that's coming from the synthesis of those very stable compounds from modern society. So that's why I went back to think of what I learned in the previous job and try to think if in my future job, how I can combine those things together and develop my own unique program. So I learn every day. Still to this day, biology is such a huge field. I don't even think biologists can claim themselves to be that for everything. Chemistry is the same. So I have to be very humble to say, I only know what I know, and I know what I don't know. This is a very famous Ramsfield quote. And there's still things I don't know.
Exactly. And I think having this interdisciplinary team really helps you continue to build this knowledge and continue to learn things every day. You were doing some really exciting work in the laboratory, and I'd love to talk about some of your current research. So, Susie, is there a particular project that you are just the most excited about at the moment that you want to share with all of us?
Yeah, sure. I think there are probably 2 projects I can share. Let's talk about the first one. That's kind of the very first idea when I came back to build my lab, is this forever chemicals. And when I was in Iowa, before that, I would study toxins like phytotoxins. Then I get to know a lot of broad range of potential threatening emerging contaminants in the environment. Forever chemicals particularly got my attention because I was on the interstate like workforce. We work with many, many other states and their state agencies and even commodity groups to find out if anything can be done, even establish a standard for those forever chemical analysis, detection, surveillance, and of course, mitigation strategy. And the answer is apparently no. I don't think at this time in our society we have a consensus like solution to fix this problem. So what I was trying to think as my future project when I go to A&M is I want to look for a hybrid between chemistry and biology. So that's I kind of moved back to what I did a long time ago, use fungus as a biological vehicle to reduce the contamination. Our hypothesis is that the fungus can degrade this chemical in a natural way. So that's why when I came back, I looked into build a fungus library, and then I work with the strains I worked with before and look for strains I have not worked with before. And then combining other technology like material science and, of course, analytical chemistry and environmental chemistry to augment the natural process, make them more efficient to degrade those forever chemicals. That's one project. And I'm very excited to continue this project because, as I mentioned, one way you can build an augment process to make the degradation more efficient. And on the other side, you can look for more powerful organisms that can degrade this compound. So the chemistry, engineering, come together with biology, and each of those have to develop and advance, then you can have the ultimate best solution.
Oh, absolutely. And I think it can be overwhelming to know where to start for some of these sorts of things. So how did you ultimately decide this white rot fungus was a good place to start? Or did you have indications that this was a place that would make sense to begin looking for these degraders of PFAS?
Yeah, it's wonderful because, as I mentioned, this is not coming from nowhere. We have to, as scientists, typically we develop ideas based on what's the observation and what are the existing evidence from the experiments. But back in 2015, 2016, I did work with scientists on a project while trying to use red water fungus to degrade toxin called dioxin. I mean, that's a different chemical. But anyway, it's a very toxic synthetic chemical produced by human beings. At that time, we put it with different fungus strain. It's well known that red water fungus can degrade very persistent chemicals because of the nature they grow on wood. And wood is composed of one of the very stable natural polymer molecules like lignin in this world. If you inherit a piece of furniture from your grandma, you know how stable they are, right? Mm-hmm. That's the evidence, and people know that. And scientists studying even more to understand how those lignins can be degraded by red water fungus. There's a lot of existing knowledge in this field. So over at that time, I was doing dioxin remediation with red water fungus. And then now I'm thinking of the forever chemical is even more hard. This chemical is composed of the most strongest chemical bond human being can form. Definitely, the natural system is not efficient enough to deal with this chemical problem as what the human society is very impatient now. You say that the Congress wants to fix the problem in 4 years and the president can fix the country in 4 years. That's impossible. And then because of this very inefficient and natural process, then I'm thinking, okay, we'll have to add some synthetic process into this picture. And that's why not only use red water fungus, I'm thinking of we have to efficiently enrich this chemical from the environment. So we have to develop a solvent, which everybody else has been doing. And then if we have the solvent, people can get rid of with the existing technology. So what I was thinking at that time, I want to give a solvent a unique function that the solvent can serve as a food for the fungus to use so that I can have one stone for two birds or maybe even more. So that's kind of the idea I thought at that time. Actually, I think it's during COVID or pre-COVID time. I don't remember.
Those years blend together.
Yes.
So this was project number one of the two projects that you mentioned you were excited about. And I think this is phenomenal work. Do you want to tell us about the second one?
Yeah. And then another question when I asked is, of course, now I understand the persistent organics are bad because we synthesize them for their desired properties. So they have to be stable. But on the environmental side, they're not very good. Then it prompts me to think of the synthesis process, right? Because those chemicals are really man-made. And how you can make that are more sustainable in the future to reduce those contamination. Think of the nature system. And a lot of previous work has been done in the biosynthesis of biofermentation as well. For example, from the World War II, some of the chemicals are primarily produced by organisms like lactic acid. So I'm thinking, okay, this microorganisms has a powerful capacity to degrade persistent chemicals. On the other side, they have this powerful capacity to synthesize useful compounds. That's kind of a collaboration with another colleague in the department. They have used the bacteria. That's the beauty of in the plant pathology and microbiology department, being a chemist. We have a professor has a library of isolates. He isolated when he was studying plant pathogen. Then, of course, there are other collaborators together. And I worked with another professor. That professor is very good at manipulating and engineering of those microbes. So when I came to this idea, I want to work with a production of chemical, use microorganisms. Those are natural my collaborators. And the idea I had at that time is, okay, CO2 is a contaminant. And CO2 was not a contaminant 30 years ago. People don't really call that a contaminant. And then the advancement in this field is people have been able to use electricity to turn CO2 into something. They're going to be more useful than CO2 because CO2 is not a contaminant. But in this sense, CO2 is not a contaminant anymore, but it's a feedstock. So what I'm thinking is, okay, I know what's state in the art that people can convert CO2 with electricity into some compounds. And then what if I can use those compounds to produce even more useful compounds? And the microbes are the wonderful thing that can do this job potentially. So that's kind of the idea of that project. And I worked with other people who supply the strain. And then the people have some modification on the strain, use different substrates. So we come together with this idea, let's go to CO2. We change CO2 into something that the microbe can use as food. This idea, if you think, is no different than the first project I talked about. But the nature is different for purpose. The first one is this chemical is too persistent. I don't like it, I want to degrade it. But this one is okay, CO2 is bad, but we can use that more useful. So we use microorganisms to change the CO2 and add a value to make that a product that's desired. But the nature behind it is still the same, right?
Right. I love this change in perspective, this sort of shift from thinking about these contaminants as being a problem that you need to fix into sort of finding this as a productive opportunity where you can use this problem that we just happen to have to make something even better and to sort of fuel something that will be valuable.
Yeah, exactly. I think that also kind of match with the current mind thinking, circular economy. So yes, you have something bad, you dispose it, you bury it, you mitigate. You don't add value to it. But if you can add value, that potentially it has more commercialization potential and people are more willing to buy.
Well, Susie, these research projects are amazing, and we really appreciate you sharing them with all of us today. And listeners, if you're looking for more opportunities to hear about some really cool science, New England Biolab's podcast, Lessons from Lab and Life, is your source for staying informed about the most up-to-date molecular biology breakthroughs. Host Lydia Morrison interviews prominent scientists and delves into their research to uncover how their findings affect society in meaningful ways. Tune in to learn from the experts and get a glimpse of the cutting-edge advancements being made by scientists around the globe. Don't miss out on this opportunity to gain new knowledge and stay informed on the latest discoveries. Tune in to Lessons from Lab and Life today, wherever you listen to podcasts. And Susie, you shared some of the remarkable work that you and your team have been doing, but I know not every project works out exactly as you envisioned. So there are lots of failures, challenges, and struggles in science. So Susie, do you have a story of one of your own failures or challenges that you can tell us more about?
I have a lot. I have even more to share than those two. So for example, when we came to that idea of converting CO2 to substrates that a bacteria can use, I want to build a hybrid system. I will tell you, in the first couple of the months, we kill every bacteria we feed into the system. That's only one example. In order to make a scientific project success, I think you have to fail. And then the good thing about a scientific approach is you have a failure because something happened as a precondition. And our job is to find out why that's happening. So troubleshooting is always what we do in our daily task. And if I'm getting a new student, the first thing I want to tell them is, okay, make sure you are prepared to fail. And then you can be success. You can have success maybe the third year of your graduate study. You have to try so many times you fail, then you get a wonderful project you want to pursue for the next 2 years. And that can happen to everybody. So that king of bacteria is only one example. Another thing is like even in our measurement, sometimes we are living in a world with so many uncertainties. So your results can go to a random error that's expected, but your result can have systematic error that needs troubleshooting. And I see students a lot of times are struggling with, oh, my test results are so bad. I tell them that's normal. You have to have an understanding of the uncertainty in your experiment. Then you can come up with a very good design, especially a control design, to control the error you're going to produce, or to eliminate the error that's huge, but you don't need to consider as important. So I think that's not a real example, but this is a principle happening with every project with a forever chemical measurement. With building the material that's going to be a solvent. You have to repeat it so many times, then finally you know, because your control is designed wrongly, then you're comparing with a standard that's not even producible.
So Susie, I have to ask, what keeps you going? What keeps you from falling into despair maybe after like the 18th time you put a bacteria in there and it all died?
Because I know there's hope. Because I see other people have published. And I know we can find a way to stop the cell from dying. But there are some projects I have to drop because after some investigation, and you have to kind of weigh out what are the odds and what are the successful rate. So it's a kind of still a risk management approach in running those research projects. And typically for many scientists, I believe it's the same thing with many other wonderful professors in their own field is you have to run so many projects and then you try, then you fail. Then you pick the one you think that's most promising with the least risk of failure. And it's hard to tell you, do you have a quantitative measurement of, okay, I know this one will work better than the other one. And I think sometimes it's instinct driven.
Oh, I love it. And I think that persistence, that perseverance, just keep getting up again after you fall down is key in science. Because like you said, there are so many failures and challenges and it's totally normal, but we don't just want to focus on the tough times. Susie, I'd love to talk about your successes as well. So Do you have a favorite success story, whether it was a big win or maybe just a small but particularly meaningful one?
All those scientific projects developing this technology for treatment and other technology for synthesis, I'm very, very excited about anything coming out. But I want to think one of the biggest success I think in my life over time to this point is actually I can work on a project that can change people's life. I don't care it's like 1,000 people or 1 million or it's each individual. As long as I see the evidence, people learn from what I learn, and then I can take actions to do something to improve that situation. So I'll give this example is the recent study that we are monitoring water quality in Texas. And then the chemical study is actually inorganic contaminant. It's not organic, different from the forever chemicals I talked about. It's arsenic. So we know in certain spots in Texas, we have this water contain high arsenic from private wells, but people don't know. You don't have that information. And then once you don't have the information, you assume, because my grandma drink it, my grandpa drink it, and my father drink it. Why should I be bothered? This inorganic contaminant is invisible. You have to test it, then you know. So what we did is actually we offer those free testing, and we solicited for the samples from the community. And then we found out, especially like in the past year, and this year, there are some pretty high spots of those inorganic arsenic in drinking water. So I contact the people and tell them what are the potential mitigation strategy you can use. And some people even seek for doctor's advice. So I think that's a real success I can testify. I see people benefit from the research we're doing.
Oh, I think that is absolutely amazing. And if you, in these cases, you found there was arsenic in the drinking water in those private wells, are there things that you can do to the wells to make the water drinkable again? Or are you just sort of treating the people who have maybe consumed too much arsenic?
So if the person has been exposed to very high, then I think the doctor's advice is much better than mine. Don't listen to me.
Right.
And then one of the most immediate things I think is really reduce the exposure, right? So what we did in this particular project, there are many things you can deal with it. But with private well drinking water, we recommend the household with some products we tested. Not necessarily have to endorse the product. We can share with them, okay, we validated for this filter, and we did an experiment in our lab. It shows it reduced this concentration after you filter with this filter. So that's kind of a very straightforward method we're using to teach people. If you want to fix it, this is the first thing you can try. And there are other more expensive ways. So again, this interview is not talking about the water quality. I'm just giving you an example of feasible and practical actions we try to teach people.
Absolutely. And I love that the work that you do has these immediate impacts that you can see, because I think that's often not the case in science. Oftentimes you sort of hope that you might see a real world impact in your lifetime, but you may or may not. So I think being able to see that, I think, is really inherently motivating and rewarding, I'm sure.
Yeah, exactly. And especially you will be inspired rather than discouraged by the non-responsive population. And we're living in a world that people are not responsive anymore because there are too many spams and misinformation. I don't even answer the phone if it's a strange number. I trust many people are like me. Mm-hmm. And then we see those people even sending letters back, telling us the address you sent, the person has moved. It inspired us knowing that people value our work. That's why they even write back with a letter. People don't even text. People don't even email. They only like push and they only click on like.
I think that is amazing. So we've talked about the phenomenal work that you get to do in the lab. And I love, like you said, that you're able to see these real world impacts of your work. But I think another thing about science careers that's absolutely amazing are the opportunities that you get to travel, Susie. So do you have a favorite place that your science has taken you?
To any audience who is not interested in science or who is very interested in science, there's so many opportunities you can travel because you have to go out and share your research, right? You have to go out and get a poster, give a presentation, talk to people, introduce your work, and learn from other people. So for many of the conferences in the chemistry side, they are in wonderful places like California, Chicago, Florida, New Orleans, any place you can go. In the past, I'm a much nerdy person and I have very poor sense of directions. So once I get out, all I can rely is my GPS. I know sometimes people say, hey, she's a female driver. Yes, I am, but don't discriminate me on my capability of arriving at my destination. So I typically go to conferences, study the people's profile and the network I'm going to build. I don't even dare to go out. But recently, I'm kind of a little bit getting into photography. And then I did take the time last time when I went to San Francisco. So I went to the Golden Bridge and take some pictures. These are examples. If you go to Florida, if you like Disney at this age, you can still go there.
Oh, I think that is absolutely amazing. And I know this is something that a lot of people in science really enjoy, being able to not only go to all these different places that you might not otherwise go yourself, but also being able to meet people from all over the world. And I think the people in science are another sort of undersold thing about the field and why it's so great. I think oftentimes there's these stereotypes of people being portrayed as very serious, very critical. And I know the research is very important, of course, but just some of the most funny and most creative people I know are scientists. And I think everyone has experienced these particular quirks and lab traditions and just funny, amazing memories that you share together. And we love to show these human side of science moments on our show as well. So Susie, do you have an example perhaps of a lab tradition that you have in your lab now, or one from a past lab, or just a funny memory with colleagues that you shared that you could tell us about that shows this human side of science?
So one of the things I like to do in my lab, I think that's a kind of more family time. So we do this journal club, then we order pizza. And then sometimes we change, we order some different food, and I find that more expensive. And we are scientists, so we are very budget oriented. And then in that journal club, we eat, and then we try to be very critical for the best paper we can pick. And it's a fun process. I enjoy that very much, and I encourage my students to not just present other people's work, but their critiques. Like you're writing a literature review, you're writing a drama review, you're writing a music review, then I don't like it.
I like that, sort of bringing the critical angle from a lot of different perspectives.
So that's what I try to encourage and use this as a not necessarily lab tradition, but I think that's important family time together. So people are more relaxed and we eat and then we chat.
I think that's amazing. I think food is a great way to bring everybody together. And I love to hear that you've got this journal club tradition in your lab, because I know you're tackling some tough questions. So creative thinking is required here. So I know oftentimes in science, you're limited, whether it's funding, staff, technology, feasibility, time, whatever the case may be. But Susie, if we took away the things that normally hold you back and we gave you everything you could dream up, what is the one research question you would want to answer most?
Yeah, still degradation of the most persistent chemicals in this world. I think we are empowered by all the technology we learned in the past, but we created so many things that's not going to go away easily. And we want to wait until the civilization disappear, and then they disappear. Because I think, as I mentioned, human beings are very impatient. We want to see things coming out more quickly. And how we can balance this impatience together with all the resources we have and make that ends meet is a very important question for us to ask.
Absolutely. So what is the limiting factor here? What's keeping you from being able to sort of crack that nut and solve what could potentially degrade these most poisonous chemicals?
Yeah, there's a nature for that chemical, right? That's because it's the strongest bond on this world. And I want to use a very mild way to destroy that. We can have a very high temperature. We can burn it up for sure, but that's not the way it's sustainable. It's very energy intensive as well. So that's kind of really, I hope to come up with a solution that can be applied in the field in the future. And then like many past issues we can fix. But again, we know for human society that many issues we probably cannot fix.
Well, I think this is a tremendously important question, and we appreciate you dreaming big with us on the show. And I'd love to end our conversation with advice if we can. So when you think back on your life and your career, Susie, is there one piece of advice that somebody gave you that really helped you that you can share with our listeners today?
Yeah, I think it's a self-indication. Because as human beings, we make mistakes all the time. And I am very proud to say I made more mistakes than the success I had. I had so few successes in my lifetime. But I keep telling myself, just be yourself and be strong. And then try to learn as much from your failure instead of be a failure and a loser in that situation. And there's this self-indication, even talk to yourself in this way will help a lot.
I love that advice, Susie. Is there any other last note of inspiration or last piece of advice that you want to leave everyone with at the end of our conversation today?
Sure, because I am a teacher and I'm a mentor for students. So I say that many very young scientists with a mind like me 20, 30 years ago, not knowing exactly what you want to do, is be humble and learn things as much as you can. Don't waste your time. Our time and our, of course, other people say our money. Where your time is where your interest and where your future is. So put much time into the thing that can help you achieve your goal is the most critical part. So make a good decision on how to use your time and what you want to learn It's very important.
I love this message. Listeners, we must make sure we are spending our precious time well every day. And Susie, if our listeners want to learn more about you and more about the amazing work that you're doing, what is the best way for them to do so?
They can check my website. I'll try to update my website more often in the future. My lab website.
Oh, that's perfect. Well, listeners, definitely go to Susie's lab website, read up on the phenomenal work that's coming out of her lab. And Susie, it's been such a pleasure to chat with you today. Thank you so much for your time.
My pleasure too. I appreciate this opportunity for sharing.
It was wonderful to hear more about you and your work. And listeners, always great to have you here with us as well. Thank you so much for joining us. And we hope we'll see you again on our next episode of People Behind the Science.